So now that we've seen the different functions that proteins can carry out, the questions that comes in mind is how can such a group of molecules that are built from the same building blocks can carry out such different functions? The answer is that despite the fact that proteins are all built from the same building blocks, in this case amino acids, they are still chemically and structurally diverse, and this determines their functional diversity. Each protein chain is made of amino acids that appear in a very specific order. We call it the sequence of the protein, or the amino acid sequence of the protein chain. All of these are amino acids, but there are 20 different types of amino acids that normally appear in proteins. And each amino acid has a different chemical and physical properties. As a result, in each chain, some amino acids are attracted to one another, while others are repelled from one another. And this creates in each chain a specific pattern of attraction and repulsion forces or interactions, which drive the chain to fold in three-dimensional space into a specific structure, a specific three-dimensional structure. In this structure, the distance between mutually attracted amino acids is minimized, and the distance between mutually repelled amino acids is maximized. And each such structure has a different function. So what's the relationship between the specific structure that each protein has and its function? When we went over the different functions of proteins, we saw that virtually all of them are based on the ability of the protein to bind a ligand. We saw, for example, that enzymes bind their substrates, receptors bind hormones or neurotransmitters, and so on. In each case, the binding between the ligand and the protein is mediated through a binding site on the surface of the protein. Sometimes it's deeper. And the binding is made possible because the two are compatible with one another, which means that the binding site has a specific geometry that is compatible with the shape of the ligand. So the two match one another like a hand to a glove. But moreover, the amino acids inside the binding site are positioned around the ligand in a way that they can favorably interact with the ligand and hold it in place. These are the non-covalent interactions or forces that mediate the binding. In the case of enzymes, some of these amino acids not only bind the ligand, but they also act chemically on it to allow it to undergo a chemical reaction and being converted from a reactant to a product. So now we have the full picture of the relationships between the amino acid sequence of a protein and its function. Each protein has a different amino acid sequence, and therefore a different three-dimensional structure with a different binding site. And this allows each protein to bind specifically a different ligand or substrate and have it and carry out a different function on it, whether it's just binding or acting chemically on it in an enzymatic reaction. We see that in these processes, non-covalent interactions or forces are central. First, as we saw, they induce the specific folding of each protein chain into a specific three-dimensional structure. And secondly, they allow the protein to bind through its binding site its specific ligand. Non-covalent interactions are very weak. They are approximately 100 times weaker than covalent bonds. But that's actually an advantage because it allows protein structures to be flexible or dynamic, and it allows proteins to bind their ligands reversibly. Here we can see a general scheme of the non-covalent interactions that we observe in proteins and in other macromolecules. And We'll discuss some of them in detail in the following slides. But at this point, I'd just like you to notice that most of these interactions are electrostatic. They involve electric charges. And this includes, for example, ionic interactions or hydrogen bonds. And there's only one type of interactions in this scheme that is not electrostatic. And in fact, it is not even a physical interaction in the strict sense. It's actually an effect called the hydrophobic effect, which results from the medium in which the two interacting species reside.